All right, just as a reminder, exam 4 is going to cover chapter 11, which is meiosis. How do the gametes actually form and where does the genetics go in terms of the DNA? Into chapter 12, which is heredity. And now the first 17 slides of chapter 14. The rest of chapter 14 and all of 15 will show up as 20 of the 100 questions on your final. The other 80 questions on the final will come from each of the 4 exams, 20 questions from each exam. You're free to come in to my office hours, which I know is inconvenient for some of you, having a night class, but you're free to come into my office hours anytime from this point to the final exam to overlook the old exams and do a little bit of study. You can do anything with the exams except for take pictures or video of them. So if you like to come into my office and spend an hour or so, writing down different questions to help you remind yourself of what you're looking at, that's perfectly acceptable. So chapter 14 focuses on DNA, and the first 17 slides are going to focus on the history of DNA. And so the first person is Frederick Griffith. Frederick Griffith was a British doctor during World War II, and before World War II he was also in the military, and he was looking for a vaccine strategy against Streptococcus pneumoniae, which is a bacterium that causes pneumonia-like symptoms. It's not the only bacteria that causes pneumonia, but it's one of them, thus its name. So his studies in 1928 gave us the first clue that DNA was involved. At this point, DNA was understood as being a molecule in organisms, but understanding exactly what DNA was and how it worked was still very much in its infancy. So Frederick Griffith's experiment gave us a lot of insight into DNA. So he had 2 strands of Streptococcus. One strain was smooth strain, which is S. This was the virulent strain. Virulent means loud, able to cause disease. So the reason it was able to cause disease is something that we'll study in microbiology in the 211 class for those of you that need to take that next. It's what we call a capsule. And so the capsule goes around the bacteria and acts like an extra layer of camouflage. But underneath a microscope, it looks like it's a nice, smooth sphere. So he called it the S strain. The R strain was missing that capsule, and so when we looked underneath the microscope at it, it had a more rough appearance, and also it just so happened to be non-virulent, meaning it could not cause disease. So he infected mice with both strands to see what happened with both strains. Now here is the controls. Remember all the way back to chapter 1 talking about science and having the need for controls. So the controls here was the live virulent and the live non-virulent. He infected mice with each. Voila. The virulent strain caused the mouse to succumb to disease. The non-virulent strain, the mouse lived. Nothing special there. Those are controls. The next one was the experiment. So he heat killed, meaning basically he probably just boiled the bacterium of the virulent strain, gave it to the mouse lived. That makes sense. We killed the bacteria. Even though we infect it with dead bacteria, bacteria has to be alive to cause disease. Now the interesting one, the actual point of his entire experiments and what actually launched his name into DNA history is this example here on the right, which is a mouse that has been infected with a mixture of the heat-killed virulent, which is the same one we see there on the left, which allowed the mouse to live, with the live non-virulent, or the strain that can't cause disease, which we also seen in the previous slide, allowed the mouse to live. So what is going on when we do a mixture of both the heat-killed and the live non-virulent that the mouse actually dies? Well, what we know now is that bacteria are very good at picking up DNA from their environment. So these heat-killed virulent, even though the bacterium was dead, its DNA still existed. And therefore the non-virulent strain was picking up the DNA from the environment, which gave it the ability to now make that capsule, and now the strain has become virulent. And so here, this was the very first instance or experiment that we can look back on showing that bacterium mixed together was able to pull the DNA from its environment and utilize it. This is what we call transformation. Hershey and Chase came along about 30 years later, 20 some years later, and they were investigating bacteriophages. Now, bacteriophages are an interesting organism, not even an organism, it's a virus that infects bacteria. And again, from chapter 1, we learned that viruses are not considered alive because they don't have a cell. But here, and a bacteriophage is a particular type of virus that only infects bacteria. Nature is weird. So bacteriophages are only made up of DNA and protein. And so this made for a very good model to use to determine which one of these was the most important, DNA or protein. And the reason why there was this argument was that DNA is only made up of the 4 nucleotides, guanine, thymine, cytosine, and adenine. Proteins are made up of 20 plus amino acids, all in different combinations. So scientists at the time could not wrap their head around the fact that a molecule made up of only 4 nucleotides was actually controlling all the genetics on Earth. They really wanted to see that it was the proteins that were responsible for genetic information, because 20 amino acids gave you a lot more work to work with in terms of diversity. So this is what they were using this experiment for. So when they did their experiment, they were using radioactive elements, which was the standard operating technique in science labs up until the late 90s, early 2000s. And what we do here is we just use isotopes, as we learned in chapter 2. The isotopes of these particular elements has an extra neutron, I believe, and thus it is what we call radioactive. That particular element doesn't like having that extra neutron, and it's going to try to get rid of it, which when it gets rid of it is something that we can actually pick up and read with different machines, one called a scintillation counter. But here, just to keep it simple, they're using radioactive phosphorus and radioactive sulfur. I didn't want you looking at that and going, what the heck? Why would they be doing that? Well, this was pretty standard techniques back in, not so far away. So it gave us a way to track the molecules. And so when we did this experiment, he has the phages that were grown with radioactive sulfur. Well, there is no sulfur in DNA. And so sulfur is only found in certain amino acids that make up proteins. So in this case, the radioactive sulfur made for a very good tracking agent for the protein. And so when he allowed the bacteriophage, which, they just have this really cool design. This is what they look like actually in electron microscope pictures. They use these legs essentially to sit down on the bacterium, and then here is basically a big syringe that they use to inject their DNA into the bacteria, and then essentially this becomes just an empty capsule, protein capsule, that then is useless. The DNA infected into the bacteria then has all the information to make new protein coats, new bacteriophages. But at the time they didn't know this. So the top one is following the protein labeled bacteriophages, and they used a blender to separate the phage coats. And what's interesting is actually at Cold Spring Harbor laboratories where these experiments were conducted, the actual blender they used is an old Waring blend, Waring being the name brand of the blender, but they actually have it on display in the museum at Cold Spring Harbor because it was such a huge experiment. So here they used a blender to separate the bacteria and bacteriophages. They centrifuged, which is just spinning at a high speed, and the bacteria, being much heavier than the viruses, will all congregate down at the bottom, which is the bacterial pellet. Up here, viruses are much smaller, so the bacteriophages are going to stay in what's called the supernatant. The supernatant is the liquid that's left behind after centrifuge. The pellet is going to be the solid mass that we can see down at the bottom. So here, when they followed the protein coat was all found in the supernatant. The supernatant was radioactive where the pellet was not. This suggests, as we already know, the protein coat for the bacteriophage was not being injected into the bacterium, and the key data is right here where the supernatant ends up being radioactive and not the pellet. Now, when we use phosphorus is used in the phosphodiester backbone of DNA, so between every nucleotide is a phosphorus, and so this is a good way to track DNA. So here they're using the phosphorus labeled bacteriophage, and the same exact procedure was followed as was done up here at the top. And what we see now is that the bacteria pellet is actually radioactive, which suggests that DNA was what the bacteriophage was injected into the bacterium. So when we look back at the DNA structure, these are things that we've already focused on a little bit in the previous chapters. I believe in chapter 3 when we looked at macromolecules. DNA is the nucleic acid, so it is one of the 4 major macromolecules with lipids, carbohydrates, and proteins. And so DNA is made up of a deoxyribose, which is the sugar right here. And what's interesting is that there's only one small change between RNA and DNA. DNA has a hydrogen here at carbon 2. And notice here how we label the carbons 1, 2, 3, 4, 5. That's going to be important in the way that DNA is synthesized. And so we have deoxyribose, which is the name of the sugar. In RNA, it has a hydroxyl, this OH, and that becomes ribose as the sugar. So really, the only difference between deoxyribose and ribose is this extra oxygen here at carbon 2. The phosphate group is over here. This is what it's going to look like when it's a nucleotide, but when it becomes when these nucleotides get attached to the DNA, this is going to be where it attaches to. The 5' carbon right here is going to attach to the 3' carbon here. So this OH and this oxygen are going to kick off as water. What's going to be left is this phosphate, and that's going to become the phosphodiester bond that we'll see here in the next slide. Oh, I apologize. Up here is the nitrogenous bases, and so we have purines. These are the adenine and guanine, or A and G. The pyrimidines are going to be the cytosine. I'm sorry, it's been cut off here. Or it's actually wrong color. If you look and you highlight that picture, you'll see it says cytosine in white, thiamine here. And then over here is uracil. And remember, uracil is only found in RNA. It's not found in DNA. So these 4 here are the ones we find in DNA: adenine, guanine, cytosine, and thiamine. Purines have 2 ring structures, so see there's a ring here and a ring here, another ring here, another ring here, so these are purines. Pyrimidines have a single ring structure, as you can see in all 3 of these. The phosphodester backbone, as I pointed out, here would be the 5' carbon. It attaches to the 3' carbon of the previous nucleotide, and so this is going to be the orientation. We always read DNA, so if you send your DNA off for sequencing to Ancestry.com or 23andMe or whatever, what they're going to be doing is reading the nucleotides from the 5' end first to the 3' end. And so if this is a guanine and this one was an adenine, just guessing, well, it's a single ring structure, so it has to be cytosine or thiamine. If this was C, the next one would be T, and that's how we would read it out to you. We always read in a 5' to 3' manner. Now, Erwin Chargaff gave us some more interesting information, on top of Griffith's experiments and Hershey and Chase's. Chargaff was trying to figure out if there's any correlation between these 4 nucleotides. And what he was able to learn is something that we already discussed, which is adenines bind to thiamines, and cytosines bind to guanines. But what he did was he actually took the genetics of many different species, dozens upon dozens of different species, and looked at their DNA and tried to sort out the ratios of A's and T's and C's and G's. And what he found was in every single organism he looked at, the amount of A's and T's were equal and the amount of C's and G's were equal. And that helped us lead to the fact that DNA is a double helix. And we'll see that more as we go through. So this added more confusion to the question, how in the heck can a simple molecule like DNA that's only made up of 4 nucleotides carry out such complex coding functions that basically is every single living thing that we can find here on Earth? Especially when the A's and T's are equal and the C's and G's are equal. So, it seems to lend even more confusion to how DNA can actually do all these. And so along came Rosalind Franklin. Rosalind Franklin was a female scientist back in the 50s, which would have been difficult at best. But she was very good at doing what's called X-ray crystallography. And so X-rays were brand new at the time as a new study technique, so we didn't quite understand the dangers of X-rays. And so Rosalind would work in her lab basically with the X-ray machine on all the time, looking at DNA. And what she was able to do is here in this bottom right picture, I can't tell you exactly how to interpret this, but this is her famous picture that led to the idea that DNA is helical. And so what she was using was the neighbor's lab next door. Maurice Wilkins was very good at being able to isolate DNA. He didn't really know what to do with it after that. So she would go and get some of the DNA from him and look at it underneath her X-ray machine. And that's where she got these data. Now, Francis Crick and James Watson were two guys that actually did not do a single experiment, but instead they used information from Chargaff, from Rosalind Franklin, and others, and frankly even stole Rosalind Franklin's data before she could publish it and call it her own. And what they did was they took all this data and they built the famous molecular stick model, which is at the front of this chapter, which was able to show that DNA was in fact a double helix. And when they did this, they won the Nobel Prize. Unfortunately for Rosalind Franklin, she was excluded from winning the Nobel Prize because unfortunately she passed away more than likely from excessive X-ray exposure. And one of the rules of the Nobel Prize committee is that you must be alive to win the award, since there is a $1 million cash reward with it too. So Maurice Wilkins, James Watson, and Francis Crick shared the Nobel Prize for the discovery of DNA structure. Now, what do we mean for it to be a double helix? What this is suggesting is that we have two strands. We have one going in 5 to 3' manner, as shown here, and then there's going to be another one next to it that's actually going in the opposite direction. It's going to go from 5 to 3, and it's going to have these these nucleotides next to each other. And so what happens is a purine has to bind to a pyrimidine. So G's and C's go together, A's and T's go together. So one strand, if we have the information for one strand, we can automatically know what the other strand has, as long as the DNA isn't mutated or damaged. So here's an example of why G's and C's can only bond with each other. That's because there are 3 hydrogen bonding sites. And A's and T's, there's only 2 hydrogen bonding sites. So if a thiamine was to bind with this guanine, there would be this missing hydrogen bond here, which is going to start adding to instability in the DNA structure. And when that occurs, over a sequence of many different mutations, eventually the DNA is going to be damaged and start leading to problems in that individual. So here's a picture of their ball and stick model here, of course digitized, but same idea. Each of these is representing different, each of these balls representing different things. Here I like the ribbon structure on the left a little bit better. It shows the 5', and if we follow this ribbon down, we'll get down to the 3' end, and then the other side would be 5 going up to the 3' end. T is always bonding to the A of the course, the other side. G binding to a cytosine, thiamine, adenine, on and so forth. We have what's called the major groove, which is the larger groove here in between as it spins, and a minor groove. And DNA is very, very geometrical. It's always 2 nanometers in diameter, and there's going to be a distance when it makes a complete turn. So basically following right here, complete turn is finished right here. That's 3.4 nanometers. And between each nucleotide is 0.34 nanometers. So again, very geometric, molecule, which is unique. So the next question is, how did DNA replicate? And so I've kind of lent you to the idea that if we know what's on one strand, the other strand, it becomes apparent because G's and C's and A's and T's only bond to each other. But there were 3 models that were proposed. One was the conservative model, which suggests that when DNA replicates, one ends up being the old DNA, and the other one is a fresh, brand new copy. That would be conservative. The old strand conserves itself and stays intact and makes a new one. The second one is what we call semi-conservative, which means that one of the strands becomes a template for the new, and the other strand becomes a template for another new. So both of the new DNAs have one copy of the old strand and a new strand, and in both of them. And then the other model was dispersive, which is just kind of a hodgepodge of old and new without any real reasoning behind it. And so the next set of experiments was to determine which of these three was correct. So Mendelsohn and Stahl did this experiment in 1958. And what they were doing is they were using bacteria. And the reason why they were using bacteria is we can go through multiple generations of bacteria within a few hours. Essentially, bacteria grown in the right conditions can replicate and create new generations every 20 minutes. And so in this case, they were also using a radioactive isotope. And so this time it was using nitrogen. Nitrogen gets incorporated into many different components of the bacterium. And so once they grow up the bacterium completely in radioactive media, they would then take a small sampling of that bacteria and transfer it into fresh media that did not have that radioactivity. And they would follow every 20 minutes, essentially each generation of that bacteria, they'd follow and they'd take samples to determine how much radioactivity was still there. And so in round 0, of course, all of it would be radioactive because that's their 0 time point, the second that they removed it from the radioactivity into the fresh. Take a sample. Of course, all of it's going to be radioactivity because it hasn't had time to incorporate in the fresh medium. After round 1, it's going to be a mixture of both old and new, so that's still going to look like a single band. And now, see, this is actually leaning towards the idea that that DNA is semi-conservative, because here we can see that it's old and new. Second round is where we actually start seeing separation. Again, old and new. So if I take one of the new strands right here, the orange one, separate it apart, and make another one, well, now that strand of DNA is going to be completely radioactivity free. Now the other one is going to have radioactivity in it, but it's only going to be half. And so notice that we now start seeing two banding patterns. We see one representing the fresh DNA, another one representing the radioactive DNA. And this was all the information they needed to prove that DNA replicates semi-conservative. There's a YouTube video here you can watch. It will help out with understanding it from an animated point of view of what they did. The semi-conservative model was supported. The other two, conservative and dispersive, were rejected. And that helped us to understand that DNA, in fact, because of how geometric it is, one strand can help us understand what the other strand is going to have, and this is what nature follows too. Alright, that is the end of what's going to be on Chapter 4. So I will leave you here, and we will pick up more of this information as we go forward in the next couple classes. Alright, good luck studying. If you have any questions, please send them out to me. I'll be available to answer questions via email. If you want to come in for office hours to look over old exams, just shoot me an email after you take a look at my office hour availability, and let me know what day to expect you, and I'll make sure that I'm there to help you look over the exams. All right, have a great Halloween, and we'll see you next Tuesday.